Hello everyone. This is about the meiosis process, which is also called as a reductional division, and also meiosis 1 is a reductional. Meiosis 2 is actually homeotypic, which is also called as equational division, similar to mitosis. It undergoes the four stages, that is prophase, metaphase, anaphase, telophase. In the middle, cytokinesis, and may or may not be, but then it undergoes interkinesis, and then prophase, metaphase, anaphase, telophase. Then it is undergoing cytokinesis. Then proper division, it leads to the four haploid cells. Then prophase is the largest phase, and it is a very typically longer and also more complex when compared to the prophase of mitosis. It is divided into five substages: leptotin, zygotin, pakitin, diplotin, diakinesis. Lepto is separation, and gradually chromosomes visible it under the microscope. Chromatin condense to form the chromosomes. Longest and thinnest also. Bead-like structure like chromosphere is also present. All chromosomes are directed towards the centrioles, so that they are giving signals for the centrioles for removal, so that it look like a bouquet shape. And also in case of the animals, in plants it is like a synegesis type, like a clamp or a knot type. Then we'll continue with the next step, that is called as a zygotin. Immediately the zygotin is called as a synaptin, also we can call it, because synaptonemal complex is been forming between the homologous chromosomes, so we call it as a synapsis. And pair of homologous chromosomes are forming, that is called as a bivalent, or also we called as a tetrad in nature. Also develops a structure called as a synaptonemal complex, which is a nucleoprotein complex, and three thick lines of the DNA and also proteins will be formed in between the homologous chromosomes. According to the Meiosis 1956, synaptonemal complex helps in pairing and also chiasmata formation, that is X-shaped formation also will be helped by this. Next, the third step is called as a paketing. It is also thick thread-like structure, and which increase the attraction of the homologous chromosomes tightly coiled around each other, and both chromatid, each chromosome become distinct, clearly visible tetrads. Also recombination nodules will develop, and non-sister chromatids of homologous chromosomes exchange will happen, that is called as a crossing over, which leads to the genetical variabilities. And exchange is happening because of the recombinase enzyme, which is having endonuclease as well as the ligase enzyme, which can cut and also which when attach. By the end of the crossing over is completed, chromosome remain linked at the sites of the crossing over. The next stage will be the diplotin, where desynapsis process will be happening. Synaptonemal complex will be removed except at the X-shaped area. Then it will be followed by the diagonal.